Hello students, welcome to today's class. I am Bernisha, lecturer from Emmanuel College of Pharmacy. Today we are going to learn about a new topic from pharmaceutic subject, that is extraction. In this video, we are going to learn about what is extraction and what are the modern techniques which is used for the extraction of plant products. Okay, let's get into the video. First, let's see what is extraction. Extraction is nothing but it's the method of removing the active constituents from a plant or animal's tissues, which is with the help of a liquid solvent, which is also known as it is the treatment of plant or animal tissues with a liquid or a solvent, whereby the medicinally active constituents like active pharmaceutical ingredients which gets they extracted from the plant materials. Here, the two words which is commonly used or mainly used in this extraction process, that is menstruum and mark. Here, the solvent which we are using for the extraction process is known as menstruum, and the insoluble materials while we are applying or while we are giving a solvent for the extraction process, some of the active ingredients may completely dissolve with the solvents, but there are some of the insoluble materials which will be remain after the extraction process. That insoluble material which remains after the extraction process is known as mark. So the solvents which we used for the extraction process is known as menstruum, and the insoluble materials that is remains after the extraction is known as mark. So extraction is nothing but it is the method of removing the active constituents from the plant material or an animal tissues. Next, what is galenicals? Usually, galenicals is nothing but it is deals with the branch of extraction. So, in old days, there is a pharmacist known as Galen. He is a Greek pharmacist. He is the one who introduced the method of extracting the cure drugs from the plant and animal tissues. So, the branch of dealing with the extraction of plant and animal drugs are known as galenical pharmacy, and the products which we are getting from that extraction is known as galenicals. So we know what is the extract. Extract is the preparation of crude drugs which consists of the pharmaceutical constituents or the chemical constituents which is actively present in the plant or animal tissues and which is soluble in the menstruum. That is the solvent we are used for dissolving the active ingredient. There are three types of extracts which is used in the extraction techniques which we get through the extraction. So first is dry extract that may be around tap or cap in like for example belladonna extract. It may a solid structure. It will be like a solid structure. The next one is soft extract, which will be like an ointment or suppositories. For example, glycyrrhiza extract. And the third one is the liquid extract, which we will be using as a tincture. So there are three type of extracts we are getting through this extraction process. One is the dry extract, and another one is the soft extracts, and another one is the liquid extract. Next is the solvents which we are used for the extraction. So solvents are the menstruum we are used in the extraction process to dissolve the active ingredient which is present in the plants. So here in extraction process, the solvents which we are employing will have the capability to penetrate into the tissues of the drug, and then it allows the active ingredients to dissolve in the solvents. So various solvents are used. Most commonly we will use the water as well as the alcohol. First we are using the water. Here the water is a solvent for protein, coloring matter, gums, glycosides, sugars, alkaloidal salts, enzymes and many organic acids as well as some of the organic salt. So if there are many active ingredients which will be dissolved in the water. So most commonly used solvents in extraction processes water. And the next one like waxes, fats, fixed oils, they may be insoluble in water. For that purpose, we will use another solvents. Here, what will be the advantages and disadvantages of water? Here, the advantages of water may be it is very cheap in nature. So, we will get availability of water is very high so that we can easily get the water for the extraction purpose. And the next, it has a wide solvent action. Most of the active ingredients are soluble in water and it is non-toxic and it is also non-inflammable. So these are the main advantages of using water as a solvent in extraction. And next one is what are the disadvantages of water? Though the water has some of the advantages, it also has some of the disadvantages. It may some of the substances which dissolves in a wide range are undesirable and may interfere with the clarity of preparations because sometimes it may cause us fermentation or decomposition of preparations. And next, the most advantages we face with the water is it helps in the growth of fungi, bacteria. So we have to add some of the preservatives to the crude drugs which we are extracted with the help of water. So this water, while we are using the water, we need to add some extra preservatives for preserving the extract for a long period of time. And the next one, the water may also causes hydrolysis in many substances. So this may also causes some of the chemical degradation of the crude drugs. So these are some of the disadvantages of using water. And the next one is alcohol. This is the solvent which we are used for the water insoluble substances. So the alcohol is a solvent of alkaloids, alkaloid salt, glycosides, volatile oils as well as the resins. So it can also dissolve the coloring matter, tannins, organic acids and salts, but it does not dissolve albuminous matter, gums, waxes, fats, fixed oils, and sugar. So it is mainly used for the extraction of alkaloids, glycosides, etc. So why we are using this alcohol? So it may have some of the advantages. So here, what is the main advantages? The fungi and the bacteria, the microorganisms cannot grow in this solvent. So there is no need for the addition of preservatives. If we are taking the alcohol with a concentration of 20% or more, there will be no growth of microorganisms. So there will be no addition of preservatives is needed. So next one is it is neutral, so that the extraction of the products will be compatible with the other products. So this solvent is compatible with the other products also. It is non-toxic in the concentration mostly present in the preparations. A small amount of heat is required to concentrate the alcoholic preparation. While we are using the water, it needs more amount of heat to make it as a concentrate for preparation. But while we are using an alcohol, we need a less amount of heat which is used to concentrate the alcoholic preparations. Next one is many active ingredients or active drugs are completely dissolved in these alcohols. Though this alcohol has many advantages, it also have some disadvantages as it has very costly. Though the solvent we are used, the alcohol as a solvent means it is very costly, and because the preparation of the solvent is also very high method, so we have to use so that it is very costly. So there will be the less availability than compared to that of water. So these are the solvents which we are commonly used in the extraction process, mainly water and another one is alcohol. Next one is what are the factors which affects the extraction process? There are some of the external factors which will affect the extraction process, mainly the nature of data. What will be the nature of the crude drug? What will be the nature of the active ingredient which is present in the plant products, which may also affect the extraction process. The next one is the solvent. The solvent we have chosen for the extraction process, which also plays a major role in the extraction process. So next one is temperature. When we are giving a more temperature to it, it will be making the active ingredient will be degraded fastly. So that the temperature also plays a major role in the extraction process, then along with pH and the particle size of the active ingredient. Some of this may easily affect the extraction process. And the next one is the methods of extractions. There are several methods which we are used for the extraction of crude drugs. Commonly, we are using infusion process, decoction process, digestion, maceration, percolation, continuous hot extraction, supercritical fluid extraction, countercurrent extraction, microwave assisted extraction, and ultrasonication assisted extractions. Here we are commonly using infusion, decoction, digestion, maceration and percolation because these are the very simple methods which is used for the extraction of crude drugs. Let's see one by one in this video. First, we are going to learn about infusion. Infusion is nothing but infusing means we have to completely soak the drug product in the water. For that, first we have to take the powdered drugs. We have to pour water over the drugs and we have to allow it to keep in contact with water for a stated period, usually around 15 minutes, and it may need some of the occasional stirring, and finally we have to filtering it off the liquids. The mark is nothing but the undissolved particle which has not to be pressed. We should not press the mark substances in infusion process. Here mainly we will use the boiling water as a solvent because because uh, this boiling water has the greater ability or solvent action than that of the cold water. Usually in infusion we have to take the crude drug product or crude drug powder and we allow it to keep it in contact with the solvent medium more than 15 minutes so that we will get a filtrate, and that is known as our extracted product. There are two types of infusions, that is fresh infusion and concentrated infusion. Fresh infusion is nothing but just we have to getting the filtrate after being allowed in contact with the solvent for around 15 minutes. But in concentrated infusion we have to do some concentration process to get a thick consistency of that infusion. For that, this we have to how we have to prepare the infusion. Here we have to take a coffee pots or tea pots mainly. Uh, First here we have to place the drug in the bottom of the pot. We have when we are taking a coffee pots or tea pots, we have to place the drug in the bottom of the pot. Then we have to add some of the hot water, and we have to stir it for 3 to 4 times during the period of infusions. And it also prepared in muslin cloth, and in this the drug is kept in a muslin cloth and you have to suspend just into the below the level of water in the beaker. So these are the simple preparation or simple extraction technique which is used to get the extraction from the crude product. First, we have already studied that there are two types of infusion. One is fresh infusion. Here we will use aqueous liquid which is used as the menstruum. Here mainly we will prepare by first we have to take the coarse drug and then we have to add a hot water to it, and as per the Indian pharmacopoeia, we will soak it for around 12 hours for the formulations. Example, fresh infusion of quassia. And the next one is concentrated infusion. In this picture, we will see that the apparatus which we have used with the beaker which we are used for the preparation of infusion. The next one is concentrated infusion, which is 8 times stronger than that of the fresh infusion. Here mainly 20 to 25% of the alcohol concentration is used as the menstruum. In fresh infusion, we will use the boiling water, but in concentrated infusion, we will use 20 to 25% of alcohol. Here it is prepared by double or triple maceration process. We will study this maceration process later. it can be stored for a longer period of time, as we are using alcohol for this infusion process there, so that we can keep this in extract for a longer period of time. For example, concentrated infusion of quassia. Next is the apparatus which we are used to for this infusion. Here we consist of a covered jar. Mainly it is made up of earthenware or stainless steel, ceramic or porcelain. So this beaker, so see that the jar, in the above the jar there is a perforated tray, so that we can upon which we can keep the crude drug, and so that we can pour the water through the perforated tray. So the plant may be enclosed loosely in the small muslin bag and suspended in the jar at a height where it will be just covered by the liquid. Sometimes we will keep the drug directly into the jar. Otherwise, we will tie it in the muslin cloth and just suspend it into the below the level of water present in the jar. Next process is decoction. We all know that the decoction is nothing but it is the similar process which is similar to the tea preparation in our daily life. So in this process, the crude drug which is boiled in a specified volume of water for a defined time, and when it is cool, we have to strain or filtered. So after boiling, the liquid is strained and the water is passed through the content strainer. Mainly, it is suitable for extracting water-soluble or heat-stable constituents, for example, tea and coffee. And the next is digestion process. Digestion process is nothing but it is the form of maceration. Maceration means it is softening process. Here we have to um, here we have to gently heat the solvent. It is like first we have studied about infusion. No, it's like infusion techniques, but when we are introducing a solvent to the product, and we have to apply some of the heat during the process of extractions. So during the process of this extraction, while we are applying the heat, the drug will slowly dissolve in our solvent. It is mainly used for the extraction of morphine or some of the active ingredients which will need the elevated temperature to dissolve the active ingredient present in it. So this is digestion. Digestion or infusion are same like process, but in this digestion we have to add the solvent to it, and we have to elevate some of the temperature during the process of extraction. And the next one is maceration. Maceration, it is nothing but in this process, the solid ingredients are placed in a stoppered container. So here mainly we have to allow it to stand for a period of at least 3 to 7 days with frequent agitations. So then the mixture is strained through sieves and it, the mark is pressed and the combined liquids clarified by decantation after standing. So it is also same as the process of infusion, but in infusion we won't press the mark, but in maceration process we will press the remaining mark also to get the combined liquids. Now let's see what is the process behind what are the steps we have to follow during this maceration process. So during this process of maceration, first we have to take the plant material which we have to crush or cut or small or moderately we have to make it a coarse powder. We have to place this coarse powder into the closed vessels. Here whole of the selected solvent, mainly in the coarse powder, we have to add the solvent. Solvent means nothing but the maybe water or alcohol. It is known as menstruum. And the next we have to allow it to stand for around 7 days by shaking occasionally. Next, we have to strain the liquid after the 7 days, and the solid residue, that is the mark, which have to be pressed so that we will recover the occluded solutions. Next, we have to properly strain the liquids and mix the both the liquids. Next, we have to clarify the substance by filtration methods, and next we have to evaporate, and sometimes we will get a concentrated solutions. Next, there are three types of macerations. First maceration technique is simple maceration. Next is double maceration, and third one is triple maceration. First is a simple maceration. Mainly, the simple maceration is for the organized and unorganized crude drug. For example, tincture of orange or tincture of lemon. And the second one is double maceration. Here we will get some of the concentrated crude drug, concentrated extractions like concentrated infusion of orange, like that. And the triple maceration, here we will carry out this process with the help of heat and stirring. So these are the three types of maceration we are commonly used in this extraction process, mainly simple maceration, double maceration, and triple maceration. Let's explain these three types of maceration in the following slides. First is simple maceration. For simple macerations, mainly there are two types. We have already studied that simple maceration is for both the organized drugs and also for the unorganized drugs. Organized drugs means nothing but the directly we will get it from the plants like roots, stems, leaves, etc. Though in our unorganized drugs, it is like some of the secondary metabolites or some of the secretions, that is like gums, waxes, resins. So some of the secretions from the plant products which is used as the crude drugs. So that is known as unorganized drugs. Like if we are using the plant part directly for extraction, it is known as organized drug. And when we are using the plant byproduct or plant secreted products for the extraction process, it is called as unorganized drugs. So first is for organized drug. Here for organized drug we have to use alcohol as the menstruum. In this process first we have the drug is placed within the menstruum and we have to close the vessel for around 7 days. We have to shake it like normal process. We have to shake it up occasionally, and after 7 days we have to strain the liquids, and the mark is also pressed so that the expressed liquid is mixed with the strained liquids. So we have to again filter it to get a clear liquid. So this is just like a simple maceration, but for a organized drug we have to add alcohol, which is used as a menstruum. And the next for unorganized drug, for unorganized drugs like gums, resins, oleoresins, like that. So in this process, the unorganized drug. So there for organized drug, we will fully place the drug with the menstruum. But in unorganized drug, we have to place it with 4 by 5th volume of menstruum. So up to a full level or up to mark 4 by 5th volume of menstruum, we have to place the unorganized drug, and we have to close the vessel and keep it for 7 days. After a period of time, we have to what the liquid has to be filtered. But in unorganized drug, we should not press the mark. The pressing of mark is unnecessary. After that, we will final volume is adjusted with the one volume of menstruum through the filter. So up to if you are taking 100 ml, we have to make up to 90 ml. We have to add up to 90 or 80 ml for filtration up to 7 days. And after filtering, we have to add the again 10 ml with it to make up the volume. Okay. If we are taking for 50 ml, we have to add 40 ml. Up to 40 ml, you have to add for the first 7 days. And after filtering that from the 7 days, we have to add the remaining 10 ml with the to make up it to 50 ml. This is for the preparation of unorganized drug. And the second one is double maceration process. Double maceration process means here the menstruum we are used here will be divided into two parts. So that in this process we have to macerate the drug twice the time. So first we will use the menstruum and keep it aside for 7 days. But in this process the drug is macerated twice, which is used in the using the same menstruum, but we have to divide it into two parts. For that, we will have some of the formulas for the volume of menstruum required for the first maceration. That is the total volume of menstruum. If you are taking total volume of the menstruum, that is around 50 ml or 100 ml. So total volume of menstruum, which minus volume to be retained by the drug to by 2 plus volume to be retained by drug. So volume of menstruum which is required for the second maceration is equal to total volume of menstruum minus volume of menstruum used in the first maceration. So what are the volume of menstruum we have to use in the first maceration should be subtracted or left to get the maceration volume, menstruum volume. We have to use it for the second maceration. Here in this process, the whole drug is macerated for 48 hours with the quantity of menstruum for first maceration. We have to strain the liquid and press the mark after the 48 hours. Then again it has to be macerated for 24 hours with the remaining quantity of menstruum for second maceration. Strain the liquid, press the mark after the 24 hours. Then we have to mix the liquids obtained from both the two maceration and allow it to stand for 14 days, and then we have to filter it. So this is the double maceration method. For example, concentrated infusion of orange and concentrated compound infusion of Sirata. And the next one is triple maceration process. Triple maceration process means it is the maceration process. Here the menstruum is divided into three parts. In double maceration process, the menstruum is divided into two parts. In triple maceration process, the menstruum is divided into three parts. In this process, the drug is macerated thrice by using the menstruum, which is calculated using the formulas. Once we have in first maceration, that is the same formula, that is the total volume of menstruum minus volume retained by the drug by 3. There we have used 2, here by 3 plus volume to be retained by the drug. Next in the second for second and third maceration, that is the same formula, total volume of menstruum minus volume of menstruum used in the first maceration for the second maceration, and the total volume of menstruum minus volume of menstruum used in the second maceration for the third maceration process. So here in triple maceration process, the menstruum is divided into three parts. In this process, the drug is macerated for 1 hour with a part of menstruum first for first maceration. Then we have to strain it, press the mark, and again it is macerated for 1 hour with the second macerate volume of menstruum for second maceration. Then again it has to be strained and we have to press the mark. Next we have to macerate it again for 1 hour with a part of menstruum which is used for the third macerations. So after that the mark is pressed lightly. Then combine these liquids and obtain for second, third maceration, and we have to evaporate for a specified extent. Then we have to mix it. Totally we have to mix it so that the liquid obtained from the first maceration and first we have to then first we have to mix the second and third macerated liquids, and after that evaporation process, then we have to add the evaporated concentrated solution to the first obtained macerated liquids. Next, through to that we have to add 90% of alcohol, which is equal to 1/4 of the volume of the finished product. Next we have to adjust the volume with water, and we have to keep and allow to stand the solution for 14 days, then you have to filter it. For example, for example is concentrated infusion of quassia and liquid extract of senna. So the in maceration techniques we have seen the simple maceration, double maceration and triple maceration. In the simple maceration we have to use the menstruum for one time along for all the 7 days. But in double maceration, we have to divide our menstruum into two times, and in triple maceration, we have to divide our menstruum into 3 times. So these are the process we have to follow during the maceration process. Mm, Okay, students, I hope you all have learned about what is extraction, what are the solvents we have to use in the extraction techniques, and what are the different extraction techniques which is used in the extraction of food drug from the plant products, and 4 different modern techniques like infusion, digestion, decoction, along with maceration. We have completely studied the 4 techniques, and the remaining techniques we will learn it in the next videos. Let's see in the next video. Thank you.